All right, so today we're going super small to unlock some huge secrets.
Yeah, smaller than you might even imagine.
Think about it like this. It's the very blueprint of you.
The instruction manual.
Exactly, like all your ancestry, what diseases you might be prone to, even little personality quirks. It's all in there encoded in these tiny little strands we call DNA.
It's really remarkable when you think about it. For the vast majority of human history, deciphering that code felt like an impossible task.
I mean, yeah, it's such an amazing concept that we can even do it now.
Absolutely, and that's
Yeah.
exactly what we're diving into today, the story of how we unlock the ability to read DNA.
And really understand all those secrets hiding within it.
Right at the heart of it all is a pretty fascinating and maybe unlikely figure.
Yeah. Unique is definitely the right word.
Kary Mullis, a biochemist,
Yeah.
who, let's just say, wasn't your typical academic.
Yeah, not the stereotypical guy in a lab coat, you know?
Not at all. We're talking about someone who found inspiration in some pretty unexpected places.
Let's just say the 1960s and things like LSD may have played a role.
Yeah, might have influenced his thinking.
So that's our mission today, to figure out how this monumental achievement happened.
How we went from not being able to read DNA to mapping out entire genomes.
Right, and we're going to focus specifically on Mullis's pivotal discovery and its really surprising origin story.
We'll be looking closely at an article that details the discovery of PCR, the polymerase chain reaction.
Which is really central to this whole story.
Absolutely.
Yeah.
So before we get into all that, you have to appreciate just how difficult it was to analyze DNA before PCR was a thing.
I mean, yeah, it's billions of these chemical letters all lined up.
Right. Think back to that idea of a blueprint or an instruction manual for a human being.
Yeah.
Unbelievably complex.
And finding a single tiny difference, like a single mutation that causes a disease like sickle cell anemia,
Yeah, that's like searching for a needle in a haystack.
or like trying to read a single license plate on a packed highway from miles away in the dead of night.
It's a great analogy because before Mullis came along, the standard way to even attempt to find these minuscule differences in DNA was a technique called the Southern blot.
Okay, I've heard of that, but honestly, I don't really know how it works.
It's pretty complex, but to put it simply, it involved cutting DNA into fragments using special enzymes, then sorting those fragments by size on a gel, which takes time, and then you transfer them to a membrane and use what are called radioactive probes to try and locate the specific sequence you were interested in.
All right, so these probes are like little homing beacons.
Yeah, exactly. They're
Yeah.
essentially short pieces of DNA or RNA designed to bind to your target sequence. And the radioactivity allows scientists to see where they've attached.
Okay, that makes sense. So it's kind of like a reverse puzzle.
In a way, yeah, but the article highlights that this entire process could take days, even weeks to complete.
Weeks. Wow.
Yeah, and it was incredibly wasteful at every stage. Plus, you're working with radioactive materials, not exactly a recipe for quick and easy diagnostic test.
It's kind of mind-blowing that that was the standard method for so long.
Yeah, and our article actually talks about how Cedus, the biotech company where Mullis eventually worked, was trying to develop faster DNA tests, particularly for sickle cell anemia, but they kept running into this problem of the signal being too faint.
Right. Too much noise, not enough clear signal.
Exactly. Cedus was really at the forefront trying to use those early tools of biotechnology to create commercially viable DNA tests.
Which makes sense. I mean, time is money.
Yeah, they even managed to get the time down to about 7 hours for a sickle cell test, but like you said, the results were just barely noticeable.
So they were getting closer, but still not quite there.
Right. This really sets the stage for Mullis's arrival, and I think it underscores just how revolutionary his breakthrough was.
Okay, so let's finally bring Kary Mullis into the picture.
Let's do it.
I mean, the article paints this really vivid picture of him as a biochem student back in the 60s at Berkeley.
It does, and he was certainly not your typical student.
Yeah, it sounds like he was more interested in exploring altered states of consciousness than going to class.
More of a free spirit.
Right, and the article actually says that his PhD dissertation had so many jokes in it that his committee almost didn't approve it.
It's funny, but it also kind of speaks to his personality and his approach to science.
That's true. He wasn't afraid to be different.
Which is interesting because sometimes those groundbreaking ideas come from unexpected places and unconventional minds.
And that was definitely true for Mullis. I mean, did you know that he actually wrote a letter to the journal Nature describing the entire history of the universe from start to finish?
I did read that, and they actually published it.
I know, he was only 22 years old.
Incredible. It really speaks to someone who wasn't confined by traditional ways of thinking.
Right. But after graduating, he kind of drifted around a bit, worked in a cardiology lab, tried writing fiction, even worked in a bakery for a while.
A bit of a winding path.
Yeah, definitely not the typical trajectory for someone who would end up revolutionizing biotechnology.
True. So how did this guy, with a penchant for psychedelics, a quirky sense of humor and experience in a bakery, end up at a cutting-edge biotech company like Cetus?
It's a great question, and the answer is actually a pretty chance encounter.
That's right, a lucky break.
His friend Tom White, who worked at Cetus, happened to walk into the bakery where Mullis was working.
What are the odds?
Right, they started talking, and White knew about Mullis's, let's just say, extracurricular chemistry activities back at Berkeley.
His talent for synthesizing certain compounds.
Exactly. So White recommended him for a job at Cetus, highlighting his skills as a synthetic chemist.
So he was basically hired for his drug-making skills.
Kind of ironic, right?
Absolutely. But you have to remember, Cetus was one of those early biotech pioneers.
Founded in 1971, right around the time scientists were first figuring out how to manipulate DNA.
Exactly, using tools like restriction enzymes. And they had this ambitious goal of creating commercial DNA tests.
Which was a huge deal at the time.
Yeah, but they were constantly hitting a wall because of the limitations of the technology at the time.
And that's where Mullis enters the scene. But his initial role wasn't exactly glamorous.
Not quite. He was basically tasked with creating those short radioactive DNA probes that were used for the southern blots.
I can't imagine that was very exciting.
Yeah, he found it incredibly tedious, slow and repetitive.
I don't blame him.
And apparently his personality didn't exactly make things easier.
Yeah, the article doesn't really hold back. It describes him as abrasive, argumentative.
Not really a team player.
There was even a mention of him jokingly threatening to bring a gun to work.
Yeah, he was definitely a character.
To say the least. But then a pretty important development happened at Cetus. They got a brand new DNA synthesizer machine.
Which was a game changer in itself.
Totally. This machine could automate the creation of those DNA probes, basically doing a month's worth of Mullis's work in a single day.
Incredible, and it really highlights the impact of automation even in its early stages.
Right, but in a way, this automation inadvertently gave Mullis two crucial things: more free time and a ton of those DNA probes to experiment with.
Yeah, which, given his less than stellar relationships with his coworkers, is probably what led him to start spending more and more time away from the lab.
And that leads us to that fateful road trip to his cabin in Mendocino County in the spring of 1983.
The moment everything changed.
He's driving along thinking about that frustrating DNA testing problem.
And boom, the breakthrough hits.
Mullis describes this moment of insight so vividly.
It's really remarkable. He talks about seeing images of these molecules in his mind.
Even when he wasn't under the influence of LSD at the time.
It's clear that his ability to visualize these complex structures played a major role in this eureka moment.
He said he was seeing blue and pink images of electric molecules.
And the idea that struck him was so simple, yet so brilliant. Instead of trying to build a better microscope to read a single DNA sequence, what if you could just make countless copies of it?
Like a DNA photocopier.
Exactly. And that's the essence of PCR, the polymerase chain reaction.
When you look back, it seems so simple, but it really was a complete game changer.
Absolutely. It revolutionized molecular biology.
Can you break down the fundamental steps for us?
Sure. So, at its core, PCR involves 3 key steps that get repeated over and over again.
Like a cycle.
Exactly. The first step is to separate the 2 strands of the DNA. Double helix. You do this by heating the sample up to a high temperature around 95 degrees Celsius.
Okay, so you're basically unzipping the DNA molecule.
Precisely. Once those strands are separated, you lower the temperature to allow short synthetic DNA sequences called primers to anneal or bind to specific regions on each of those separated strands.
Okay, so the primers are kind of like little guides.
Right. They're designed to flank the specific DNA segment you want to copy, and they're oriented in a crucial way. They point towards each other across that target region, like 2 arrows highlighting a particular sentence in a book.
So they're saying start copying here.
Exactly. And then comes the third step, the extension phase. This is where an enzyme called DNA polymerase comes in.
I've heard of that.
Its job is to attach to where those primers have bound and then extend those primers, building a new complementary DNA strand using the original as a template.
Okay, so it's like a tiny little construction crew.
Yeah, it grabs free-floating building blocks called nucleotides and adds them one by one to the end of the primer, creating a new strand.
Now, the article mentioned something about directionality here, something about a 5' and a 3' end. Can you explain what that means and why it's important?
Absolutely. DNA polymerase can only build a new strand in one direction, from the 5' end to the 3' end.
Okay, so it's like a one-way street for building DNA.
Exactly. And because the two strands of DNA run in opposite directions, Mullis realized that he could design two different primers, one for each strand, oriented so that the polymerase would extend them towards each other.
I'm following you. So you have one primer on each strand, and they're both pointing inwards towards the target sequence?
Right. So the polymerase extends them until they meet in the middle, effectively copying that segment in between.
That's so clever.
It is. And here's where it gets really mind-blowing. After this first cycle, you now have two copies of your target DNA segment. And if you repeat the process heat to separate cool to anneal primers, extend with polymerase, each of those two copies will be copied again, giving you 4 copies.
Okay, I see where this is going.
And then 8, and then 16. It's exponential growth.
So from a single molecule, you can create billions of copies in just a few hours.
Precisely. It's like a chain reaction, hence the name polymerase chain reaction.
Now it makes sense. And the article says if you repeat this process about 30 times, you end up with over 1 billion copies of that specific DNA segment.
That's right. That tiny license plate on the moon is suddenly a giant flashing billboard.
And suddenly, something that was almost impossible to detect becomes incredibly easy to see and study.
Exactly. And the real beauty of PCR is that it's incredibly versatile. You can basically amplify any DNA sequence as long as you know the sequence on either side of it.
So human, bacterial, viral, it doesn't matter.
It really opened up a whole new world of possibilities for research and diagnostics.
It's amazing to think that something so groundbreaking came from such a simple idea.
And the article actually says that Mullis's reaction to figuring this out was to pull over his car and jot down the idea on the back of a gas receipt.
A gas receipt.
Yeah, talk about a eureka moment.
But when he excitedly brought this idea back to Cetus, the reaction wasn't exactly what he'd hoped for.
No, not everyone was immediately on board.
The article says that some of his colleagues thought the idea was too simple.
Like if it was that easy, someone would have thought of it already.
Right, and others were probably just skeptical of Mullis himself, given his, history of unconventional ideas and his not always so rigorous lab practices.
I mean, he did have that probationary period.
Because his initial attempts to get PCR to work were, well, a bit messy.
Let's just say his data wasn't the most convincing.
Right, so he was given a year to prove that PCR actually worked, and importantly, he wasn't working alone. Other scientists at Cetus were assigned to help him, and by the spring of 1985, after a lot of effort and teamwork, they finally had definitive proof.
It was a collaborative effort.
Right, and the sickle cell diagnostic test using PCR worked beautifully, and it could be completed in under 10 hours.
A huge improvement over those days, or even weeks required for the Southern blot.
So imagine that going from taking weeks with radioactive material and barely getting a signal to what Mullis and his colleagues achieved.
It's a pretty dramatic shift.
A major breakthrough, for sure. But there is still one pretty significant limitation with the original PCR protocol.
Right, the original process involved those repeated cycles of heating and cooling, remember?
Yeah.
And the DNA polymerase they were using came from E. coli bacteria, which can't withstand those high temperatures needed to separate the DNA strands.
So what did they do?
Well, after each heating cycle, fresh polymerase had to be added manually.
30 times.
Can you imagine? It was a huge time suck and really expensive.
A major bottleneck, for sure.
But then a completely unrelated discovery ends up solving this problem in a really amazing way.
It's like something out of a movie.
Back in 1964, a microbiologist named Thomas Brock and his student Hudson Freeze were exploring Yellowstone National Park.
What were they looking for?
Well, they were studying these vibrant hot springs, and they discovered a type of bacteria called Thermus aquaticus, or TAC for short.
I've heard of that.
These bacteria were thriving in these incredibly hot environments, temperatures that would kill most other organisms.
Wow, so these were some seriously tough bacteria.
Exactly. And that meant that their enzymes, including their DNA polymerase, had to be heat stable.
That's amazing.
It was a fascinating discovery. But at the time, it seemed like purely basic research. No one could have predicted how important it would become.
Right, until Kary Mullis came along.
He read about Brock and Freeze's work and immediately saw the potential for PCR.
It's like all the pieces of the puzzle just fell into place.
Right. The article actually quotes a scientist at Cetus named David Gelfand, who purified the TAC polymerase and tested it in PCR. His reaction was It worked like a charm. The Holy Grail had been achieved.
For the Holy Grail, that's quite a statement.
It was a huge breakthrough because Taq polymerase not only survived those high temperatures, it actually functioned best at those temperatures. Yeah, and the higher temperatures also increased what's called the stringency of the primer binding. Remember with the E. coli polymerase? They had to lower the temperature a lot for the primers to attach, which meant they could sometimes bind to the wrong DNA, leading to messy results.
Okay, like a lot of background noise.
Exactly.
Yeah.
But Taq eliminated that issue. You get these clean, highly specific amplifications.
There's a cleaner signal.
Precisely.
Mm.
And suddenly PCR could be completely automated. You could set it up, walk away, and come back a few hours later to billions of copies of your target DNA.
So this discovery of a heat-loving bacteria in Yellowstone revolutionized a revolutionary technology.
That's science for you, always full of surprises.
But with such a significant breakthrough, there was bound to be a race for patents and recognition, right?
Absolutely. CETUS knew they had something incredibly valuable.
Right.
Other companies were starting to take notice, so they were under pressure to patent and publish their findings quickly.
And that's where the drama starts.
Right. They urged Mullis to publish his own paper first, but he delayed for whatever reason.
Maybe he was just too busy celebrating his genius.
Maybe. But the rest of the group ended up publishing their PCR research in Science, with Mullis listed as the fourth author.
I bet he wasn't happy about that.
He was furious. He felt like they were trying to steal his credit, and he struggled to get his own paper published in any major journals. But he did eventually become the public's face of PCR, especially after a presentation he gave in 1986, and of course he won the Nobel Prize in Chemistry in 1993.
So in the end, he got the recognition he deserved.
He did, but there was definitely some controversy. Some of his colleagues felt he didn't adequately acknowledge the collaborative effort at CETUS.
It's a classic tale of scientific discovery. You have the brilliant mind, but then also the messy realities of collaboration and credit.
It happens all the time. But regardless of the drama behind the scenes, the impact of PCR has been undeniable.
Truly transformative.
Revolutionary is really the only word for it. PCR has had an impact on virtually every field of biology and medicine.
I mean, the article lists so many applications. DNA, cloning, vaccine development, detecting cancer and HIV.
Forensic science, it's been used to both exonerate the wrongly convicted and catch criminals.
It's even used to reunite families and analyze ancient DNA.
Think about Jurassic Park. That whole concept was based on the idea of amplifying ancient DNA using PCR.
Mind-blowing.
And of course, we can't forget its crucial role in COVID-19 testing.
Right. It's hard to imagine modern biology without PCR.
It really is one of the most powerful tools we have.
But the story of Kary Mullis takes another interesting turn after he wins the Nobel Prize.
Yeah, he became known for some pretty unconventional views, let's just say.
The article really paints him as a paradoxical figure. On the one hand, you have this brilliant imaginative mind that revolutionized science. But on the other hand, he had these really out-there beliefs.
Like alien abductions.
Yeah, and he was distrustful of mainstream science and had some pretty controversial views on things like global warming and HIV/AIDS.
And some of those views are actually quite harmful.
Right. The article specifically mentions how his HIV denialism had a really negative impact, especially in South Africa.
It's a reminder that brilliance in one area doesn't necessarily translate to sound judgment in others.
It's a shame, really.
But I think one of the most thought-provoking takeaways from this whole story is the connection between Mullis's journey and the increasing role of automation.
That's interesting. What do you mean?
Well, remember how his routine job of making those DNA probes was automated?
Yeah,
That basically freed up his mind to think differently, to come up with PCR.
that's a good point.
It makes you wonder if the automation of these routine tasks, even the ones we find boring, could actually create the mental space for more creative, groundbreaking ideas.
Like we're freeing up our brains to focus on the big picture stuff.
Exactly. And the article poses this question: could the rise of AI and other forms of automation lead to even more of these kinds of breakthroughs by freeing up human minds?
It's a fascinating question.
It is. Because if you think about it, maybe the future of innovation isn't just about building smarter machines, but about how those machines can help us think more creatively.
By taking care of the tedious stuff.
Right. Of course, the article also points out that this kind of freedom needs to be coupled with a mind that's trained in problem solving to really take advantage of those moments of insight.
So it's not just about having free time, it's about what you do with that free time.
Exactly.
So to bring it all back together, we've looked at this seemingly impossible task of reading DNA, the limitations of early techniques, and then the arrival of this totally unconventional biochemist who, on a random road trip, came up with this brilliant way to copy any DNA sequence.
It's a classic underdog story in a way.
Right, and then we dug into the science behind PCR, the crucial role of those heat-loving bacteria from Yellowstone, and the incredible impact this discovery has had on science and medicine.
And of course, we couldn't ignore the complexities of Kary Mullis himself.
Yeah, a brilliant mind, but also a flawed individual.
And I think the real takeaway here is that the fundamental aha moment was that shift in thinking from trying to decipher a tiny piece of information to just making so many copies that it becomes impossible to ignore.
Like going from a whisper to a shout. So for everyone listening, I want you to think about this, where in your own life, are things becoming automated? Could that free up some mental space for you to have your own unexpected breakthroughs?
Are you ready to take advantage of those moments?
And if you do have a great idea, do you have the tools and the mindset to really make it happen?
It's something to think about.
It makes you wonder what incredible discoveries are just waiting to be made.
And by whom?
Maybe someone just like you.
This deep dive into PCR and its origins has really shown us just how unpredictable scientific progress is,
Right?
and how these seemingly simple but revolutionary ideas can change the world.
And that's something we should all be excited about.
Absolutely. PCR is still evolving, and it serves as a powerful reminder that the next major scientific leap could come from anywhere.
And anyone.
Maybe even from someone listening right now.
Okay, well, that is about all the time we have for today.
It's been a pleasure.
As always, until next time, stay curious.
And keep exploring.